Hello everyone, welcome back to the sessions on the glycolysis and biochemistry. So in the previous presentations we saw the detail of glycolytic reactions where the glucose was broken down to form two molecules of pyruvate. The glucose went through the preparatory phase to produce two molecules of glyceraldehyde 3-phosphate, and each of the glyceraldehyde 3-phosphate molecules it underwent the what you call the payoff phase to produce two molecules of pyruvate. And during this whole process, we can see that two molecules of ATP, here you can see, were produced, was used in the preparatory phase. Glucose was broken down to two molecules of glyceraldehyde 3-phosphate. Each of the glyceraldehyde 3-phosphate, it went through the payoff phase to produce two molecules of pyruvate. And during this payoff phase, each of the glyceraldehyde 3-phosphate resulted in the production of one NADH and two ATP molecules. Okay, so during the production of two pyruvate molecules, two ATP was used up, two NADH molecules were produced, four ATP molecules were produced, and two water molecules were released. So this is the whole complete reaction what happened in the glycolytic pathway. So balancing the whole equation, we get glucose plus 2 NAD plus 2 ATP plus 2 inorganic phosphate, resulting in the formation of two pyruvate molecules, two molecules of NADH and H plus, and two molecules of ATP with release of two molecules of water. Okay, so this is the overall equation. Now, when you look into how many ATP molecules are produced, like how many ATP molecules are produced as a result of a single glycolytic pathway, we can see here two ATP molecules are already produced. Then one NADH is equivalent to three ATP molecules. Okay, almost three ATP molecules. So what happens is 2 NADH is equivalent to 2 into 3, 6 ATP molecules. So altogether, we can say when a single glucose molecule is getting broken down to two molecules of pyruvate in a glycolytic pathway, almost like 8 ATP molecules are being produced. So net result, it is 8 ATP molecules in a glycolytic process. Okay, so it is 8 ATP molecules are synthesized in glycolysis in aerobic condition. Okay, now coming to the fates of pyruvate. Okay, pyruvate is getting converted to different products based upon in which condition the cell is in. Okay, the glucose, it undergoes the glycolytic 10 successive reactions of glycolysis to produce two pyruvate molecules. The pyruvate molecule, on the other hand, when it is in anaerobic condition, it can undergo two different kinds of reactions. In yeast, we can say, we can see that in what you call yeast and in some bacteria like Xymomonas, the pyruvate, it undergoes alcoholic fermentation. Okay, so alcoholic fermentation is the anaerobic degradation of glucose to ethanol. Okay, glucose by glycolytic pathway producing pyruvate to ethanol. So that is the whole process is known as a alcoholic fermentation, where it is an anaerobic breakdown or anaerobic degradation of glucose to form the ethanol. And here you can see that along with that, carbon dioxide is also being produced. So under anaerobic conditions or hypoxic conditions in yeast and in certain bacteria, you can see the alcoholic fermentation taking place. On the other hand, in erythrocytes, in skeletal muscles which are undergoing rigorous activities, or in certain bacteria like lactobacillus, streptococcus, etc., you can see that there is yet another pathway being undergone, that is the lactic acid fermentation. The lactic acid fermentation or the lactate fermentation is the anaerobic degradation of glucose to form lactic acid. Okay, so these two pathways we'll be looking in detail in the coming slide. Then we have is in the aerobic condition what happens. In aerobic condition, the pyruvate, it undergoes decarboxylation to produce acetyl coenzyme A, and the acetyl coenzyme A will enter the citric acid cycle or the Krebs cycle to produce carbon dioxide, water, and a good amount of ATP during the process. So this happens during the aerobic condition. Okay, so this is yet another cycle where glucose is getting completely broken down to carbon dioxide and water with the release of good amount of ATP molecules. So this, it is referred as the Krebs cycle, which we will be looking in detail in coming presentations. Okay, the next one we have is the fermentation. Okay, these two process, it is known as a fermentation, that is anaerobic respiration is what is referred as a fermentation, right? Here the first one is the ethanol fermentation, that is the degradation of glucose to ethanol through pyruvate. Okay, so glucose to pyruvate we have already seen under glycolytic pathway. Now this is pyruvate. The first step in the ethanol fermentation is the pyruvate getting converted into acetaldehyde. It is a decarboxylation process, so carbon dioxide molecule is released, and the process is being, this particular reaction is being catalyzed by pyruvate decarboxylase. I hope you can get how the name has come, the enzyme name has come. This particular enzyme, it catalyzes a irreversible reaction of pyruvate getting converted to acetaldehyde, and this particular reaction requires thiamine pyrophosphate, the TPP, which is a transient carrier for the acetaldehyde group, and also the presence of magnesium ions. Okay, then the acetaldehyde, it will undergo, you can see here, reduction reaction. Okay, for the reduction reaction to take place, there should be a reducing agent, and in this particular reaction, it is NADH acts as a reducing agent. So NADH, it undergoes oxidation to produce NAD plus, and during this particular process, it will give away its hydrogen atom, and this is being taken up by the acetaldehyde to get itself converted into ethanol. So acetaldehyde undergoes reduction with the help of NADH molecules, and ethanol is being produced. So I hope you remember that during the process of glycolysis, the glyceraldehyde 3-phosphate undergoing oxidation and phosphorylation. During that process, NAD plus was being used up for the production of NADH, and that NADH will come over here and act as a reducing agent, and hence giving back the NAD plus, which can be used for the glycolytic pathway to keep on to be continued over there. Okay, so this is a reversible process again. Alcohol, the reaction catalyzed by alcohol dehydrogenase, it is a reversible reaction, dehydrogenase. Okay, so pyruvate, it undergoes decarboxylation to produce acetaldehyde, which undergo reduction to produce the ethanol, right? So this is ethanol fermentation. Okay, and in the presence of oxygen, especially in the case of yeast, these yeast can oxidize this alcohol into carbon dioxide and water. But in the absence of oxygen, what happens is this conversion will continue and alcohol will get accumulated, and this may later inhibit the growth of the yeast, the replication, the reproduction of yeast, because of the increased concentration of alcohol in the system. Okay, so this is about the ethanol fermentation taking place in the yeast. The next one is the lactic acid fermentation, that is the production of lactate or lactic acid from the pyruvate molecule. So as we can see, the pyruvate molecule, it undergo reduction to produce lactic acid. Okay, C double bond O has got converted into CHOH. Again, here also you can see NADH is a reducing agent. NADH is readily gives away its hydrogen atom to produce the NAD plus, to become the NAD plus oxidized form. Okay, this reaction again, it is a reversible process, and it is being catalyzed by lactate dehydrogenase. Okay, so the lactate dehydrogenase enzyme, it will convert pyruvate to lactate. It will reduce, lactate dehydrogenase will reduce pyruvate to lactate with the oxidation of NADH molecule. Okay, lactic acid dehydrogenase. So again, as we have already mentioned, the NADH, which is being produced during the oxidation of glyceraldehyde 3-phosphate in the glycolytic pathway, it is being oxidized back to NAD plus, and this NAD plus can be used for, it can be used for the glycolytic pathway, and hence it can uninterruptedly, it can continue with the process. Okay, now what happens to the lactate? Lactic acid fermentation usually takes place in the skeletal muscles, as we have already mentioned, in the vertebrate skeletal muscle cells. So this accumulation of lactic acid in the skeletal muscles, it can actually affect the normal cell function, because it is acidic in nature, so the lactic acid. So it will reduce the pH of the skeletal muscle cell, and the cellular pH is reduced to a very low level, and thus it can adversely affect the normal functioning of the cell. Okay, so the usually what happens is, if you apply, like, if oxygen is available, the lactic acid will get broken down and it will be removed from the system. Okay, but the oxygen supply has to be uh, like in skeletal muscles, what happens is, the oxygen supply, if it is continued later, the lactic acid can be removed from the system, and it can revert back to the aerobic respiration. Okay, so this is about the lactic acid fermentation. We completed the aerobic ethanol fermentation as well as the lactic acid fermentation. Okay, I hope it is clear, and you have to learn the glycolytic complete glycolysis, the 10 reactions taking place, the enzymes, the various what do you call cofactors which are involved in wherever the ATP is being produced, substrate level phosphorylations taking place. You also have to study the fates of pyruvic acid and the fermentation, the two different fermentations, the reactions as well as the enzymes involved. Okay, fine. Thank you.